Hi, I'm David and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. In question 1a, you have a transmission electron micrograph showing a section of a human liver cell. A looks circular and seems to have two layers of membrane, as you can see two lines there. You can also see the foldings of the inner membrane. So an organelle that has double membranes and folded inner membrane, it must be mitochondria. B is a membranous structure. It looks like some kind of thread in the cytoplasm. You can also see black dots on the surface. The black dots are ribosomes. So this is rough endoplasmic reticulum. Remember that when you're asked to identify or name an organelle in paper 2, do not use short form. RER, rough ER are not good enough. Please write the full name all the time. Question 2 is about the enzymes attached to the membrane of smooth ER. You have to suggest the advantages of having enzymes attached to the membrane rather than free in the lumen. To answer this question, you have to first know the functions of smooth ER. There are two main functions. Number 1, to synthesize lipid. Number 2, for detoxification. The question is asking you why it is good to have enzymes attached to the membrane. So two simple points are, these are the enzymes needed for those functions. However, there's a third point needed. So you need to talk about why having enzymes on the surface is better than having them floating around freely. First of all, it's not quite possible to have just one enzyme needed for those functions that we mentioned just now. So it is logical that many different enzymes that work together for the synthesis of lipid are all found on the membrane. Secondly, these enzymes are held together closely so that they can catalyze each step in the pathway. And the last point is similar to the concept of immobilized enzymes. When enzymes are attached to a membrane, they have greater stability due to the support they have from the surface that they attach to. B. Explain the advantages of using a transmission electron microscope compared with a light microscope when viewing a liver cell. When you want to compare these two microscopes, there are three things that you should always mention. Number 1, which one has a greater resolution? Number 2, what are the values of maximum resolution for the both of them? Number 3, which one can show a greater detail when magnified? So here are the points. Firstly, electron microscope has a higher resolution. You can also use the definition, which is the ability to see objects that are close together as separate objects. Then give the value for the maximum resolution, 0.5 nanometer versus 200 nanometer. Because of this higher resolution, electron microscope can use higher magnification without loss of detail. So, it has a greater ability to see more organelles, for example, smooth ER ribosomes. You can also name any other organelles which you can only see under electron microscope. They do have 1 point for AVP in this question. For example, the preparation of specimen is very thin so that a greater internal detail can be seen under electron microscope. In question number 2, you have a diagram of the human circulatory system. A1, state why the human circulatory system is described as a closed double circulation. When you want to define a term made up by two words, you should define each of the words separately. So the best thing to do is to make a sentence for each of the words here. Closed circulation is because blood is contained within blood vessels. Double circulation is because blood flows through the heart twice for one complete circuit of the body. You can also name the pulmonary and systemic circulation for the second point. Pulmonary circulation is the one where blood travels from the heart to the lungs, then back to the heart again. Systemic is when blood travels from the heart to the rest of the body, then back to the heart again. Next, you have to identify blood vessel Q and blood vessel R shown in the figure. We need to use the arrow to know the direction of the blood flow. Q brings blood back to the heart, so it must be a vein. It is connected to the right atrium, so it is the vena cava. R transports blood away from the heart, so it's an artery. It is connected to the left ventricle and brings blood to the whole body, such as head and arms and the liver. So R must be aorta. 3. You need to describe the differences between the structure of blood vessel Q and the structure of blood vessel R. But in your answer, you should not refer to valves. Since they are vena cava and aorta, the largest vein and artery in our body, what you have to do is to state the differences between these two types of blood vessels. Vein has a thinner wall as it does not need to withstand high blood pressure. It also needs to allow skeletal muscle around them to push and squeeze the blood inside them. so the pressure can increase and blood would traverse back to the heart. Due to the thinner wall, their lumen is wider in relation to the thickness of the wall compared to the artery. They do not have a thick tunica media, hence they also have less smooth muscle proportionately, as it is the main type of tissue found in tunica media. Lastly, they do not need to be as elastic as artery, so they have fewer elastic fibers. In B, you have the structure of an antibody molecule found in the blood plasma of a shark. You can see that there are only two chains held together by some bonds in between them. Question 1. State how the quaternary structure of a human antibody molecule differs from the quaternary structure of the shark antibody molecule. To answer this question, you need to know the structure of human antibodies. Each molecule contains 4 polypeptide chains, 2 heavy and 2 light chains. Since quaternary structure is defined as a protein that consists of more than 1 polypeptide chains, the difference in the number of polypeptide chains in these 2 antibodies is the answer. Question 2 talks about the use of these antibodies in cancer treatment. You are asked to suggest how using the smaller shark antibodies may be more effective in reaching a greater percentage of cancer cells than human antibodies. When you inject antibodies into a person's blood, it is going to travel in the body until it reaches the capillaries. Endothelial cells of capillary wall have gaps between them. The antibodies will pass through the gaps and end up in the tissue fluid. Then they can bind to the antigens of the targeted cell and destroy them. So, being smaller in size, shark antibodies can pass through the gaps more easily, and more of them can bind to and destroy cancer cells. Another thing is, injection of antibodies into a person's body can possibly cause immune response, since it is not something produced by the person itself. The smaller shark antibodies have a lesser chance to trigger an immune response compared to the larger human antibody. There is 1 mark for AVP as well. For example, the shark antibodies can easily diffuse or move through tumor to reach more cells. Question 3 wants you to explain how injection of antibodies into the bloodstream can protect a person from disease after infection by a pathogen. The injection of antibodies into a person's body is an artificial passive immunity, as the person does not produce the antibodies him or herself. It can give an immediate protection, since we don't have to wait for events like clonal selection and clonal expansion to take place. Right after the injection, antibodies can bind to the antigen on the pathogen. You can also use the specific role of antibodies to answer this question. For example, they can act as antitoxin. It means they can make a toxic substance to become harmless. This is known as neutralization of toxin. They may also enhance phagocytosis. This is because antibodies can first bind to the antigen on pathogen, then their constant region can bind to the receptor on the cell surface membrane of the phagocytes. The other specific roles of antibodies are under AVP, so you can get maximum 1 mark by mentioning any one of them. Question number 3 is about carbonic anhydrase. A1. Explain why this protein is described as globular. The command word in this question is explain. It means that you have to provide reasons in your answer. The first reason is it has a spherical or ball shape. Secondly, all globular proteins are water soluble. You can also provide elaborations why they are water soluble. It is because amino acids with hydrophilic R groups are on the outside of the molecule. The fourth point is the role of globular proteins. It has physiological or metabolic role. This means that they involve in some physiological or metabolic processes in our body. And the last point, carbonic anhydrase is an enzyme, and all enzymes are globular proteins. Number 2, state the function of carbonic anhydrase in red blood cells. The word state here means that you just have to provide straight to the point answers. Elaboration in detail is not required. So you just have to mention the reaction catalyzed by carbonic anhydrase, which is the reaction between water and carbon dioxide. Carbonic acid will form. They also give you 1 mark if you state the reverse reaction which occurs in the lungs. In B, you have a diagram of a small section of DNA molecule. The first question is, you have to identify the base X and state the evidence that supports this identification. Base X has 2 rings, so it must be a purine, either adenine or guanine. There are 3 hydrogen bonds between base X and its complementary base pair. So this must be CG base pairs, as AT would only have 2 hydrogen bonds. So base X is guanine, and the reason is it has a double ring base and there are 3 hydrogen bonds with the complementary base. There's one AVP mark for the detail of the difference between the structure of adenine and guanine. Question 2 talks about a mutation that results in the deletion of base X, which affect the polypeptide produced. You have to explain how this deletion may affect the polypeptide produced during protein synthesis. Now, let me show you how deletion of a base can affect the polypeptide coded by the gene. First of all, genetic code is a triplet base. Every 3 bases code for 1 amino acid. If one base is deleted, when the DNA is transcribed, the mRNA will have 1 base missing. All the codons in the mRNA from that point onwards is now changed, as the reading frame is no longer the same. This is called a frame shift mutation. When a polypeptide is produced from this mRNA, all the amino acids added after that point of mutation is altered. Now let's go through the marking scheme. First, mention that every 3 bases codes for 1 amino acid. The deletion of 1 base leads to frame shift mutation. This causes a change in the mRNA codons. Every triplets or every codons after the point of mutation will be different. So, the primary structure or the sequence of amino acid in the polypeptide will be altered. This may also introduce a stop codon. If this happens, you will have the production of a shortened polypeptide. There is one mark for AVP. For example, if you mention that the mutation can cause an error in RNA splicing. In the last question, you are asked to suggest the effect of a gene mutation in an intron. To answer this question, you have to know the difference between introns and exons. Introns are the non-coding parts of our DNA or RNA. They are removed during post-transcriptional modification of mRNA. Some of them have regulatory function where they control the expression of other genes. So, if there's a mutation in an intron, it may have no effect on the protein structure because they are non-coding. However, they may affect RNA splicing. This may result in a non-functional or reduced function protein being produced. If this intron has a regulatory function, the mutation may affect the regulation of gene expression. You can also get this third point by giving any specific example. In question number 4, you have a diagram showing you virosome which is used in some vaccines against influenza. The first question is that you have to state the differences between the structure of a virosome and a virus. First of all viruses must have genetic material, either RNA or DNA, but virosome in this picture doesn't have it. Another structure all viruses must have is the protein codes called capsid. Lastly, the envelopes of phospholipid membranes only exist for some viruses. Not all of them have it. Question 2. Explain how the structure of the virosome suggests that the central area of the virosome is aqueous. In the picture, you can see the hydrophilic heads of the phospholipids are pointing inwards to the center of the virosomes. If it is not an aqueous environment, you would have the fatty acid tails pointing that way instead. Because of the heads of phosphate group of phospholipids are hydrophilic and they are pointing towards the center of the virosome, so it must be aqueous. In B, you have some information about hemagglutinin, which is used in a vaccine. The question here is, suggest why hemagglutinin is present in virosome used in the vaccine for influenza. When we use a molecule in a vaccine, that molecule acts as a non-self or foreign antigen to the body of the receiver. This non-self antigen will trigger or stimulate the immune response. You can also say that it provides an artificial active immunity. They will give marks if you can give some details of the events that happen when immune response occur. For example, macrophage perform phagocytosis to present antigen. B or T lymphocytes are being activated in the process of clonal selection. And lastly, the most important event we want to have is the formation of memory cells. C talks about different strains of influenza. You have to suggest why the primary structure of the active site of neuraminidase remains unchanged in each strain of the influenza virus. There are 2 ways to answer this question. The first one is to explain in terms of enzyme structure. The 3D shape of an enzyme's active site determines its function. So, if there's no change in the shape of active site, then the enzyme can bind to the same substrate. So the function doesn't change. This ensures that even when the virus mutates, it can still use this enzyme to leave the host cell and infect other cells. The second way to answer this question is to use the genetic basis of protein synthesis. When the virus mutates, as long as the mutation is not occurring in the gene codes for neuraminidase, or it doesn't affect the amino acid sequence making up the active site, the enzyme can still function normally. The gene that codes for this enzyme is essential for the survivor. So the viruses are smart enough to know that, however they mutate, they cannot change the gene coding for this enzyme. In question 5a, you are asked to complete a table with information about the two pathways water can take when crossing the roots to the xylem tissue. The first one is when water moves through plasmodesmata. Since plasmodesmata connect the cytoplasm of adjacent plant cells, this statement is telling you that water is moving in the cytoplasm. The answer is Sym+. The other pathway is Apo+, where water moves via the cell walls and through the intercellular spaces. In B, we have a photomicrograph of a stem. The question is, you have to draw a label line and the letter T to identify xylem tissue. First of all, look for the vascular bundles. You will find xylem inside them. Xylem vessels are the largest cells in a vascular bundle, so those large circles are the xylem vessels. Together, they are the xylem tissue. C talks about a vacuole that releases hydrolytic enzymes to the cytoplasm. You have to name an organelle found in animal cells that has a similar function to the vacuole in the developing xylem vessel elements. There is only one organelle with a membrane that can release hydrolytic enzymes in animal cells. It is lysosomes. Question 2 asks you to suggest how the permeability of the tonoplast changes and explain how this change could result in the rupture of the tonoplast. There are two things you need to do here: suggest and explain. Let's give the suggestions first. To cause a membrane to rupture, you need water to move in. So, it is very likely that the cell has an increased permeability to water. To achieve this, one common way is to have more aquaporins, the protein channels which allows movement of water. Now, let's give the explanations. There must be a water potential gradient for water to move this direction. So, the water potential in the vacuole is lower than that of cytoplasm. Due to the increased permeability and the water potential gradient, more water enters vacuole by osmosis. Vacuole increases in volume or pressure until it bursts. 3 talks about the pH change in the xylem vessels and how this activates enzyme. You have to suggest how a change in the pH of cytoplasm can activate enzymes. To activate an enzyme, we want it to be more able to bind with its substrate, so it can catalyze the reaction. The pH change may cause active site becomes more complementary to the substrate. It is because pH change can affect the R group interactions between the amino acids, for example, hydrogen and ionic bonds. This slightly altering the shape of the enzyme, hence its active sites. Sometimes an inactive enzyme may have a blocked active site. If we remove it, it is activated and can start binding to the substrate. The next point is not something we talk about often in the syllabus. There is something called cofactor in our cells. They help an enzyme to work. So, pH change may lead to the association of a cofactor with the enzyme, making it functional. D describes a disease called Pierce disease. The question is, explain why Pierce disease can be described as an infectious disease. To explain this, you need to link the definition of infectious diseases to the example given here. Firstly, the question says it is caused by a bacteria. All infectious diseases are caused by pathogen. Secondly, infectious diseases can be transmitted from an infected individual to an uninfected individual. In this case, from one plant to another. Thirdly, diseases bring harm to the infected individuals. The question has told you the negative impact of this condition, so it fits the definition of a disease. Question 2 wants you to state the type of organism that caused malaria and name one of the organisms. Malaria is caused by a type of Protoctis. Then, give any one of the four names. Take note that the genus name must start with capital letter and species name does not. And since you can't really isolate your handwriting to show that they are scientific names, you should underline those names. 6a shows you a photomicrograph of plant cells in stages of the mitotic cell cycle. The first question is, you have to name the stage of mitosis shown in cell Q. You can see the chromosomes are separating and moving towards the opposite poles. So, this is anaphase. 2. Outline the roles of mitosis in a healthy plant. When the question says outlines, it means that you don't have to explain in detail. So, just provide 2 rows of mitosis in a plant, then you will get the 2 marks. Firstly, is to increase number of cells for growth. Secondly, repair of tissue by replacement of cells. Take note that it's wrong to say that it's for the repair of cells, because that would mean the repairing of DNA in a cell, which is not a role of mitosis. Next, new cells can be made to replace all dead, damaged or worn out cells. It is also for asexual reproduction. A general statement to state that mitosis can produce genetically identical daughter cells can get you 1 mark as well. In this question, you are given a graph showing the results of an experiment. Cancer cells are treated with different concentration of a drug. The percentage of cells in stages of mitosis and the ratio of the number of cells in anaphase to metaphase are being shown here. From this graph, you can see that as the concentration of the drug paclitaxel increases, percentage of cells in stages of mitosis increases, but the ratio of number of cells in anaphase to metaphase decreases. Now you have to describe the results and suggest an explanation for the effect of paclitaxel on the mitotic cell cycle. First of all, let's do the description. You just have to make 2 straightforward statements to show how the ratio and percentage change with the concentration of drug increases. Then, quote in data to support your points. For example, as the concentration of paclitaxel increases from 5 to 50 nanometer per dm cube, ratio of cells in anaphase to metaphase decreases by 0.23. After that, let's come up with some explanations. The ratio shows that more cells are in metaphase, indicating that they are stopping in that stage. That's why we see them more than anaphase. You can also suggest the mechanism for halts in metaphase. For example, the centromeres do not divide, or spindle fibers are prevented from being shortening. That's why they do not move on to anaphase. That's all for today. If you think my videos are useful, don't forget to like, comment and subscribe. If you have any questions, feel free to ask me at the comment section. Thank you for watching and see you again soon.